Great. Welcome, everyone. Just a friendly reminder to please turn off your video during the talk. Today, we're very excited to have Dr. Grace Aldrovandi to talk to us and give us an update on all the exciting clinical work that's happening here at UCLA. Grace did her MD degree in Montreal at McGill University, followed by residency in pediatrics at Montreal Children's Hospital. She's a physician scientist with over 20 years, very impressive experience designing and carrying out laboratory-based studies and also clinical work to understand HIV pathogenesis in the US and Africa. And she has very impressive track record of NIH funding for all these studies for decades. And she received the Elizabeth Glaser Scientist Award for contributions to this field. Her lab is really a powerhouse in understanding, for example, innate and adaptive immunity in infants that are born to HIV infected women, and also breastfeeding, the effects of breastfeeding, and also microbiome perturbations in infants that are born to these infected mothers. And she was a professor at University of Alabama, Birmingham, followed by USC before she became directors of clinical microbiology laboratory and clinical virology laboratory at Children's Hospital in Los Angeles since 2010. And she's also professor of pediatrics and chief of pediatrics at Mattel Children's Hospital here at UCLA. She's had many leadership roles in multiple NIH-funded clinical trials networks, such as the AIDS Clinical Trials Group, International Maternal Pediatric Adolescent AIDS Clinical Trials, and the Adolescent Trials Network. So needless to say, she Grace really has exceptional expertise, and no one is more perfect than her to shift into SARS-CoV-2 research in terms of testing and serological assays for COVID, which I think she will give us all the updates today in her talk. So Grace, thank you so much for taking the time, and we look forward to your seminar very much.
Thank you very much, Melody. And I'd like to start off by thanking you all for inviting me to give this update. And I should say that this is really, I'm speaking on behalf of what we call the UCLA COVID-19 Emergency Research Network. And way back in, whoops, why is it not advancing? It worked before.
Did you try, maybe, are you clicking on the, what about
there, I got it.
Sometimes it's stuck, yeah.
Okay, so I'm speaking on behalf of a fantastic group of colleagues, and I know a number of you share with me the disorientation that this pandemic has brought to us all. It is now September the 9th. And a lot has happened, but to bring you back a bit. So to give you an idea, as an infectious disease physician, we not only have to keep up with all the changes that we're dealing with in the clinic and in the hospital, but also with the infodemic. And you can see that really since about early May, there have been over 2,000 publications on SARS-CoV-2. And I love information. I'm a bit of a junkie, but I've now reached the point that I'm not sure that Darwin was right. But I'd like to thank the organizers just for having these conferences, because I think it's important that we try to keep up in this field that increases almost as quickly as the virus itself. So back in March, a group of us got together because we realized that winter was coming for the Game of Thrones fans, that there was this pandemic looming, and we wanted to position ourselves in UCLA to be able to study it and make a contribution. And so Annie Remoyne, Jen Fulcher, Nicole Tobin, Otto Yang, Paul Krogstad, and others, we got together and we sort of sketched out projects that we thought would be important and divvied up the spoils, if you will. And I'm going to provide an update on the projects that we have. I've spoken to the previous incarnation of this group about our health services worker and first responder study, which is the largest study in our portfolio. And I have the great pleasure of working with Annie Remoyne from the Fielding School of Public Health on this project. Jen Fulcher, who's an adult infectious disease physician, has been leading studies looking at hospitalized patients. Nicole Tobin and Paul Krogstad have been looking at SARS-CoV-2 in breast milk. Otto Yang, again, adult infectious diseases, has been studying persons, and now they've gone down to children and adults who have recovered from their SARS-CoV-2 infection. And we call that study the CLEVER study. And then Patty Gantz and Ashley Gray. Ashley is an extraordinary fellow in pediatric hematology oncology, are leading a really interesting study trying to understand the role of children in the spread of SARS-CoV-2 within households, within immunocompromised people. And I'll talk a bit about that. What I won't talk about was a spin-off study, and this was looking at COVID-19 in pregnant women. And Yalda and Karen both have different cohorts, and we've worked with them on some of the laboratory studies, looking at the placenta and amniotic fluid, and we have one paper in press and another submitted, and many more studies to come. But I won't talk about that because it's a little bit outside our initial emergency research network. So the largest study is the UCLA Health Services Worker and First Responder study, as I mentioned to you. And we got approval to do this study March 25th. And what we had wanted to look at was asymptomatic healthcare workers and first responders. And it was a difficult time for the health system for us to approach them because they had just been hit. If you remember, the governor on the 19th had put in the shelter-in-place orders, and there were a number of healthcare workers who were off for a variety of reasons. And here we were going to the command center saying, hey, we want to identify your asymptomatic healthcare workers in this study. And they were wonderful. In the end, we came up with a compromise that allowed us all to learn. And the design is really quite simple. We see healthcare workers every 2 weeks. We take saliva, we do a mid-turbinate swab collection, and we draw blood. We ask them a bunch of questions, including mental health questions. And they have their blood drawn once a month, and the other studies are done every 2 weeks. They're followed for a year. If they're infected, sampling, and that's been quite interesting. We had to do this at a time, well, you will remember, there were shortages of PPE. We had to maintain social distancing, and the return to work policy was something that was evolving and involved considerable thought. What we wanted to do was to understand the epidemiology of this new pandemic. And in particular, I remind you at the time, there was still debate as to whether there was such a thing as asymptomatic infection and spread. Very interested in the virology, the shedding, comparing mid-turbinate swab versus saliva collection. At the time, reinfection was on our list. But to be honest, it was more theoretical than real. A number of us were very interested in the immunology of this new pathogen. And we were also very interested in the mental health aspects of being in a pandemic. And this has been our touchstone, our guiding principles are really reflected in this slide. What at least I didn't realize at the time when we were doing the healthcare worker study and firefighters is that they're really quite distinct populations. Now, when we set up our healthcare services study, we really wanted to include more than just the frontline healthcare workers in the hospital on the COVID ward. We wanted to design something that would allow us to also look at community prevalence. So our inclusion criteria were really, you just have to work at UCLA. And we did try to target environmental services, dining halls, staff, others who might be exposed but not traditionally thought of. But we also have many participants who work in offices who don't have patient contact whatsoever. I went over the schedule of visits. Now the logistics were and continue to be a little bit challenging. But it was much easier to do it here at Ronald Reagan. And we had employees coming on and booking appointments. So even though that was a lift, it wasn't quite as challenging as working with the first responders. And the first responders we have been working with are firefighters. And there we're looking at the actual firefighters themselves. And as if you will, a control or another flavor in them is also we are enrolling their dispatchers. So the dispatchers sit in the command center and dispatch fire engines and EMT services, but they are not in the front line. So their risk would be more community than actual contact with infected persons. Because of the logistical challenges, we only see our first responders once a month. But we do, again, take the same samples. But again, we can't, LA County is huge. Actually, for pandemic and emergency service purposes, Los Angeles County is considered a state. And so we had to have a very decentralized approach. And we don't see them every day. Our healthcare worker study, we now, we see them 5 days a week, and we now have 2 locations, one in Santa Monica and one in Reagan. But for the firefighters, we go to different places, and I'll show you, and we do it in 3 sequential days. So this is where we started. This is the garage outside the CTRC, where we first saw our healthcare worker study. This is a car loaded up with supplies going to the firefighter stations. So here's a map of Los Angeles County. You can see how big it is. The stars are where we go. And what we do with the fire department is that they arrange for different battalions to come during the 3 days that we're there. And we'll see anywhere between 60 and I think 110 people at a time. They don't have appointments. We don't know who's coming in, and it is out in the open. So our team was out there on Labor Day. And so the weather can be a little challenging, but really rewarding. So here you can see our star people for this study. This is infectious disease fellow, Rachel Martin Blaze, and this is Ashley Gray, a HEMONC fellow. And they have really stood up our firefighter study in a remarkable way. And working with the firefighters, we soon became aware that, you know, their exposures are quite different than we see in the healthcare setting. They're walking into people's homes, into people's cars. It's not controlled. They don't know what they're going to find. And the other big difference is that they work 10 days a month. Is their standard routine, but they actually live together for 24 hours. So it's a much more intense experience and more opportunity for spread. Economically, it's a little bit different too. We've run into, so we had negotiated with the health system about when people would be off if they were infected, but don't forget, we're asking people who feel perfectly well to get tested for this. And so they will be off work, and a lot of them, in addition to their 10 days, work at other stations and rely quite a bit on this moonlighting, if you will, at other stations. So there is a strong economic disincentive to get tested. The other thing they became aware of is that they, within their communities, they're known to be higher risk for having COVID. And one of them, firefighters that I was talking to, who was infected, it was somewhat poignant that, you know, their kids aren't allowed to play with other kids, and people in the neighborhood really tend to shun them, which I think is a little bit different than the average healthcare worker at UCLA. And then also their attitude towards research. They tend to be more skeptical. And I think this is where I really have to give kudos to Rachel and Ashley, because they're there, they answer their questions, and we have worked extremely closely with the leadership of the fire departments, and have quite a tremendous success in speaking to these firefighters and engaging them in our research. So we all know that fire season is upon us. This is from yesterday's LA Times. And what happens when they have these big fires is that the firefighters are called at a moment's notice. They all get into a plane or trucks or whatever, and they go into these areas, which are all triple digit, extremely dangerous, lots of equipment. And then at night, they'll go and live in a camp. And before they go to the camp, they're loaded on these trucks, etc. Quite a few people at a time. And so social distancing is not possible. And you might imagine that it's pretty difficult to ask people to keep a mask on 24/7 when you have triple digit temperatures and you're just living in a camp. And so this is a very high risk for COVID acquisition. And we are submitting today an amendment, well, a new study that to actually test folks that are going out on these strike and urban search and rescue teams before and after they go out. We were approached not only by the fire department, but LA County Department of Public Health. Logistically, this is going to be a bit challenging. So anybody from the command center, I'm going to be going to you asking how we can get access to the helicopter pad, because they're probably going to bring us the samples by helicopter. We're going to try to turn them around very, very quickly and get them the results. Challenging, but I think really, really important, because if you live in the LA area, you realize how vulnerable we are with fire. And if you watch TV or social media, you can see how climate change and our dry climate are really making fires a major threat to our lives and well-being. So we are very honored and happy to work with these individuals in any way that we can. So I was talking about logistics. This has been a really, really heavy lift. Although some of us had been involved in clinical trials, we did not have a clinical trials infrastructure. We built it all ourselves under very challenging conditions. The easiest part, which wasn't easy, was setting up the lab assays. As you know, this is, the FDA has an emergency use authorization process. I won't get into that, but because of shortages, et cetera, we had to be nimble, and we set up and validated 3 different platforms. In terms of serologic assays, and again, when we started this, there was no serology. You know, on April 16th, Florian Kramer from Mount Sinai on Twitter posted that he had a very good assay, and within 24 hours, no MTA, no nothing, he sent us reagents and his protocol. And that really helped us get off the ground running. We've now moved to a commercial assay, Abbott, which targets not the spike, but the N protein. So there are advantages to that, not the least of which is throughput. Then to see all these people, there's a ton of swabs, reagents, bleach, ethanol. And again, those have been on shortage, and there still are shortages. Tips are an issue. Freezer boxes. Oh my God, who would have thought freezer boxes are in short supply, but they are. And so this is something that we struggle with daily. And then seeing all these individuals, you have to label the tubes. And once the tubes come, you have to process and aliquot them. So if you remember, you know, once a month, we're getting 2 tubes, one which we're going to have to FICOL for our cellular assays, sera as well, and then the swabs and the saliva, et cetera. So those are 5, 6 items that have to be labeled, because we can't have a mix-up. And then they get to the lab, and that was a fair amount of work. You come to the lab and you never know what you're gonna find. So one morning, this pallet was there, and you can't see there was another pallet behind there. So when we would get stuff, we really got it. And it was challenging where you're gonna put it. Here you can see some of the kits that we're putting together. So the whole lab, all the office space was just consumed with the, and still is, the logistics of this study. But we have an absolutely phenomenal team. Here again, you can see another piece of equipment coming in. We've had to remodel our BSL-2. Again, more goodies. It's like Christmas every day here. But none of this would have been possible without the extraordinary effort of volunteer postdocs, faculty. Unfortunately, we weren't able to have graduate student volunteers, but we called ourselves Team Fauci, and I cannot say enough good things about all the labs that volunteered their effort, their equipment to help us start this lift. So what did we accomplish? Well, as of a couple of days ago, we had enrolled 2,009 participants. We've enrolled over the last couple of days, so we now have over that. 1,151 health services workers. We have a waiting list of people who want to join of over 150. We decided a while back, I honestly can't remember when, that we were only going to enroll higher risk individuals into our health services study. So that's why we've had to curtail it. We've enrolled over 850 firefighters. We've done over 8,000 RT-PCRs. We get about 200 samples a day for this project. We've done over 5,000 serologies. And we've really built an infrastructure capacity for over 2,000 PCRs by the end of this week. We anticipate by January, it'll be over 3 times that amount. We're going to be involved in other NIH studies and have building capacity for that. We now, with our Abbott, can do 200 serologic tests in 2 hours. And so that's much better throughput than we could with Florian Kramer's assay, which wasn't really high throughput in our hands. We have 2 individuals in our group who are certified to work in the BSL-3 and have been doing neutralization and culture studies. And in addition, we have validated a quantitative PCR. So who have we enrolled? And here I'm going to show you on these pyramid plots, profiles of our participants. Here you can see in blue is the UCLA healthcare worker study, red are the firefighters. And you can see how different they are. So 70% of our participants at UCLA are female, 90% of the firefighters are male. Here is the age distribution, and you can see that at the two ends of the curve, UCLA has a little bit more than the firefighters. In terms of race and ethnicity, we're still a predominantly white cohort, a few more Asians in our UCLA cohort than the firefighters. And in terms of ethnicity, more Latinx in the firefighters than in the UCLA cohort. And what have we seen so far? So one of the things that we are very interested in looking is exposure risk, even though it's kind of moot when you have community prevalences like we have in Southern California. But we asked folks if they had taken care of a COVID positive patient in the last 14 days. And you can see that our less than half in either cohort had, but you can also see that the first responders, they're not sure. And again, reflecting the uncertainties that they have when they go into somebody's house and have to do CPR or take them to the hospital. In terms of our PCR positivity rate, we're just under 1% for our health services workers. And anecdotally, I think most of these folks, if not all of them, have not got it occupationally. I think it also really speaks to how PPE works. First responders, much higher incidence. Seroprevalence, 3.5 for our health service workers, almost double that for the first responders. Interestingly, in our healthcare workers, we've identified 11 people that we call silent seroconverters. So these are people whose PCRs were negative every 2 weeks, but have serologic responses to spike, and really pretty large increases and persistent. So we are trying to understand those responses. The other thing is just last week, we have our first potential reinfection in a first responder. He was positive in June, asymptomatic, did well, but just last week he developed symptoms and is now PCR positive. So we're working with the health department to sequence and to see if he is another case of reinfection. But you can see how having a prospective study, we have samples from on these, a number of these people before they acquired their infection and are getting them relatively acutely in infection. We also partnered with Nita Quadir. It was an absolutely wonderful partnership. She worked with the CDC IV study to look at high-risk healthcare workers. And so we participated and helped her with the logistics. And that's great. And it's a sort of collaboration we've always been open to. So what are we doing currently? We're continuing to monitor and enroll high-risk individuals. We're starting our study to really look at more in-depth into their immune responses, particularly their cellular immune responses. We're, as I said, expanding the testing of the firefighters. We hope to get that through the IRB soon. And the other thing that's just again come up recently with the firefighters is long-term effects. What we have been told, and we have a call this afternoon to help them think about this, is that a number of these firefighters who had SARS-CoV-2 infection, most of them, minimally symptomatic, pass all the physical tests to get back into the field. And, you know, this isn't just having some doc in the box, listen to your lungs. They have, you know, to do so many laps and lift so many pounds, etc. However, when they're going out into the extreme conditions of fighting fires in the environments that I showed you earlier, they're having a lot of trouble. So that's concerning not only for the individuals, but you might imagine under the extreme conditions that they're fighting these fires, if one of your companions isn't able to perform optimally, that puts everybody at risk. So stay tuned. And the other thing that we want to look at is mental health data. You know, the first slide that I showed you about people being disoriented and how challenging it is for everybody. And, you know, now we know so many people with young children are at home, working from home with, you know, in small houses and stuff, and not being able to go out and interact. I mean, I'm Italian. I was meant to hug people. This is not normal times. And so with the questionnaires that we have, we really would like to look over time at how people are coping with it. So that's it for the healthcare worker studies. Now I want to focus on Jen Fulcher study, and she was interested in looking at the hospitalized patients at UCLA. And so she really wanted to get a well-curated biospecimens and also be able to use these to interrogate disease pathogenesis. She got this approval March 24th. Our healthcare workers got approved March 25th. So she beat us by a day. But again, you know, UCLA was shut down the 17th. So you can see that we were starting these projects at very challenging times. So Jen set up a REDCap database that included all these really important variables. And I think critical to her success is that she realizes that mining the EMR is best done manually by people who understand the disease and lingo, and not just relying on what's ICD codes. And importantly included specimen information. And that proved to be invaluable for treating physicians. Don't forget, nobody had ever seen this disease before. And so it was really useful for ID clinicians to ask, okay, what are we seeing in our cohort for inflammatory markers X, Y, and Z, and a number of other important clinical features. This was all entered into a database, and you could look, and you didn't have to rely on your memory. And she, as a credit to this, you can see, and I've just listed here and won't go into depth, that there are 4 publications already submitted, and she's participating in the International BMI COVID Consortium. I think you all know that being obese is a tremendous risk factor for a bad outcome when you encounter this pathogen. She also collected the biospecimens. Here they are. She had to label her tubes. I can't tell you how many Friday nights at 7 o'clock people were in the lab labeling tubes. It's prosaic, but it needs to be done. And her study was really the first clinical data and specimen collection cohort established at UCLA. And the CTSI COVID registry, you know, used her framework in its approach. And also the NIAID impact cohort study led by Elaine Reed were able to learn from her framework and what she had set up. Jen is now immunophenotyping using mass cytometry cells from folks that were hospitalized at UCLA with various degrees of disease severity. She's looking at their B cell and T cell, particularly Treg responses. She's worked with David Wong in dentistry, who I think gave a talk here a couple of weeks ago, for an improved salivary diagnostic, not only for RNA, but antibodies as well. And that's going really well. Mapping CD8 T cell responses during the course of disease, working with Quen on innate gene transcription and regulation, again, in the different flavors of the disease. And then looking at plasma, cytokine, and biomarkers and how they vary in the different disease states and with obesity. So that's Jen, stay tuned. Now I'll just talk about something dear and near to me, and this is SARS-CoV-2 and breast milk. Now, this is sort of a boutique niche area, but breastfeeding is essential for the survival of children all over the world, but particularly in less developed settings. And COVID has disrupted breastfeeding. And here I'm presenting an analysis where folks are modeling how decreases in breastfeeding will lead to death. And you can see here that a decrease in the relevant prevalence of breastfeeding, if it's only 5%, it'll mean 16,500 children will die just because they didn't get breast milk, okay? Or if you look at more severe case, if we have a 50% decrease in breastfeeding, you'll have almost 140,000 children die. Lack of breastfeeding is associated with tremendous morbidity, including stunting, which has effects on cognitive development as well as growth that will carry on for years. So this is not a fashion choice, breastfeeding. It's really a matter of life and death for children. Now, babies can acquire their SARS-CoV-2 from their moms through their breathing, contamination of surfaces, and we were most interested in breast milk. Masks really work, and it's recommended that all breastfeeding women wear masks, but what we were really interested in and what the field was interested in is, can it be transmitted through milk? To show you that, you know, the estimates and the effects on breastfeeding are not just theoretical. Here in this BMJ, they were looking in India, where they noted in some hospitals, breastfeeding has decreased 40 to 50%. And to remind you, even though the United States is #1 in terms of the number of cases, in terms of the 5-day moving average, you can see that we're coming down and India is taking off. India's got over 1 billion people, and children in India critically depend on breastfeeding. And you can see if you look at the top 10 countries, you know, other than the United States, these are settings where breastfeeding is critical. I have to say that, you know, in times of challenge, you see the best and the worst. Dannon and Nestle have been incredibly aggressive in marketing their formula and telling women that they shouldn't breastfeed because of COVID. And in my mind, this is another sin of Mark Zuckerberg, because it's really being propagated on social media. Just to give you an idea, formula is a $45 billion industry. These companies are always trying to get into developing country markets. So it's extremely challenging for those who care about the lives of children and global health. So to help address this, we set up and validated an RT-PCR assay using the Abbott EUA, with input being not only RNA, but our cultures. And then we set out to ask simple questions as to whether SARS-CoV-2 could replicate in human milk. And this work was done by Paul Krogstad in the BSL-3 here. And I'm just showing you 3 representative milks. These is on vero cells, and you can see untreated, You can see with SARS-CoV-2, you have tremendous cytopathic effect. Milk, interestingly, which had no effect on the viral cells, and it can be actually toxic to cells. And when we spiked the milk with SARS-CoV-2, there was no real difference in cytopathic effect, or when we went and put these samples on our PCR machine in the amount of virus that we were able to detect. So having the tools to be able to study this turned to, the other thing we wanted to look at was the effect of freeze-thaw, because all the milks we're going to get will be frozen. And as you know, for some viruses, freeze-thaws can decrease the amount of virus. We did not see that. And the other thing is Holder pasteurization, which is what's used by milk banks. And this was really, all the milk banks in the country were clamoring to see if what they were doing worked. And we were able to show that this worked. But as I said, with these tools, turned to collaborator at UCSD, Lars Bode, who had been collecting milk from women, and is still collecting milk from women who had SARS-CoV-2 documented infection by PCR. And these are the first 18 subjects, and you can see that all of them but one was symptomatic. These samples were collected between March and early May. So this was a time where it was extremely difficult to get testing. And right over here, you can see that we had 64 samples, and it varied tremendously from women. Only 2 of the babies were found to be SARS-CoV-2 positive by PCR. They both did well. One was negative, and then there were 2 kids who, infants who did not get tested, but clinically probably had the disease. I will point out that one of the subjects who donated milk was intubated on ECMO and received remdesivir. Her infant was positive, and we had 12 samples from her. None of them, interestingly, had detectable SARS-CoV-2, nor could we culture. On this slide, I'm showing you breast milk samples relative to the time the mom was tested. In blue is when the women were asymptomatic. In red is when they had symptoms. And you remember that in this cohort, there was only one woman, participant 18, who was asymptomatic throughout. In these 64 breast milks, we only found 1 sample that was RNA positive. Interestingly, we had a milk from this woman 2 days before she developed symptoms, and about a week after she had symptoms, and then even after that. So we had 4 samples in her. None of them were RNA positive, and we were unable to recover virus from this sample, which is not really surprising. I won't get into PCR positivity versus infectious or culture positive virus. Here's one of the first studies looking at it. But just because you have detectable RNA doesn't mean that you have infectious virus. Since then, we're about to complete over 100 milks on 75 women. We've detected 3 additional RNA positive milks, but again, none of them have grown, and they have low quantities of virus, which is consistent with that. So where are we going with this? We have, before the pandemic, we were collaborating with Galit Alter and Carolyn Ateo at the Reagan, and they had looked at some of our milks, comparing immune responses in milk and blood of women who had received different influenza vaccines. And this is from a manuscript we haven't gotten around to submitting, but we saw very distinct compartmentalization, and breast milk provides an opportunity to look in a mucosal compartment. So we're now collecting blood with our colleagues in San Diego, and we'll be doing these sorts of as Galit calls them, serum, system serology in the milks and blood of these participants. So stay tuned for that. Now I'd like to switch to Otto Yang's study, the CLEVER study, and this was first results were put into the New England Journal in July. And Otto has been following folks who have contacted him, contacted UCLA after they had their infection, and he's been studying their antibody responses. I will say that Otto has bled most of these individuals. You know, one of the things you learn in medicine, more junior faculty don't know how to draw blood. So Otto has been bleeding his subjects. I've literally gone to homes of folks who are in isolation and had to draw their blood there. So these data really are Otto's, because he's bled these individuals. So here, he gave me these slides. Unfortunately, he can't be here. This is showing you levels of the receptor binding domain, IgG, M, and A. They're different between the sexes, and I think you all know that if you're male, you do much worse with this infection than if you're female at all ages for reasons that aren't clear. Antibody levels were similar across age groups. And again, you all know that this is a disease that disproportionately knocks off older folks. However, when he looked over time, and this is his first cut of the data in the first 34 individuals that he had longitudinal samples on, he saw this rather rapid decrease in these antibodies. So he's got much more on this and cellular immune responses coming out, and I'm not going to steal his thunder on that. But again, very exciting and I think important data. And then last but not least is a study that is near and dear to my heart, even though I have not had much oxygen to devote to it. And this is the role of children in the transmission of SARS-CoV-2 in households of immunocompromised persons. And if you remember from the first slide, this is something that Patricia Gantz, an adult hem-onc, and Ashley Gray, a fellow in pediatric hematology oncology, are leading. And it's had a slow gestation just because of the logistic and other issues in enrolling families in the time of COVID. What we're going to be doing is comparing the incidence of SARS-CoV-2 infection in households that have children and households that don't have children. And our index case, in air quotes, will be immunocompromised people followed at UCLA who are at risk. We've set up home collection and questionnaires, and kudos to Ashley, because she was one of the first studies at UCLA to enable e-consent capabilities via REDCap, which was really critical for this study. And we've really only been going in the last couple of weeks, and she's already enrolled 8 family cohorts. So I think this is going to be quite interesting. It'll also offer the opportunity to look at infection across households and generations. So that's a quick summary. There's a lot of work, and I want to acknowledge and thank everybody. I'll start with COI, Annie Ramoyne, on the health services worker. This was, you know, her inspiration. And also she has just been extraordinary in her ability to not only visualize this study, but just as important, or maybe more important, to share her vision with donors and raise money that has allowed us to do this. I have to give a special shout out to folks in the lab. I always thought we worked hard until this came about. It's just been incredible. And Daisy Contreras took a research lab and turned it into a CLIA certified lab while running all these samples and validating the assays. And everybody else in the group has just been extraordinary. Thanks to Annie, we were able to hire Kathy Furbus as our scientific administrator. It's just been a thrill to work with Kathy. She loves to solve problems, and she must really love her job, because honestly, throughout this journey, we have been hit with roadblocks and challenges. It seems like almost every hour, and she just dusts herself off and says, we're going to solve this, and she does. Faith Landsman has been just instrumental in coordinating all these visits. And at the beginning, we didn't have the best sort of database. I mean, we went into this study Congo style, and when you have hundreds of people wanting to join your study, and you have to arrange their appointments, and we have to have all the tubes and everything labeled and bring them to the garage, etc. She's been extraordinary, and also in talking to participants and helping educate them. So big hats off to her. Pamina, India, and Amy were really instrumental getting this off the ground, particularly in terms of our mental health and other questionnaires. We have now study coordinators and phlebotomists, and we're so grateful for that. Otto Yang's lab has been tremendous in assisting and as scientific collaborators. The same with Jen Fulcher's lab. Scott Kitchen's lab, I have to give a special shout out for. They really stepped up and volunteered and processed so many of our samples, including the IV samples. And even after folks were able to open up their labs, they continued to volunteer their services. And that was just tremendous. April Pyle and Michael Hicks really got the award. They really wanted to help out, and we felt really bad that because of shutdowns, etc., it wasn't the easiest path to get them to help us. But hashtag they persisted, and hashtag we're very grateful for their support. Similarly, Marta, Alex, Quen, you, Sherry, Risa, and Judy Courier had all at critical times in our study stepped up and helped us. Have to give a special shout out to Harry and his lab. They have taken our lab in and have just been instrumental in giving us the room we need to breathe. Within the School of Medicine, Doctors Martin and Smale and Whitty, we have a thank. I particularly would like to thank Dr. Smale, because I know it seems like every day I was adding more people, because as we grew, we needed to them, but he was always very prompt in his responses, and we're very grateful to them. The command center, has been very helpful and supportive of us, and I appreciate how that presented challenges for them. But we are grateful, and I think our participants are. Laura Bensiker, I didn't even know she existed, and I can just not say enough good things. Whenever we've run into roadblocks, she has swooped in like our guardian angel and helped us see through this. So whoever set up Laura and hired Laura, you were just prescient. The CTSI was really helpful in the beginning, particularly when we were using their space. We've now moved into the 200 building in Santa Monica, and again have to thank Laura, Kurt, Samantha, and Sarah Santos, who runs the 200 building space, have been tremendous in letting us run our study within this space. And we recognize how challenging that is. Digital marketing helped us get up the website, another long story. Medical and dental student volunteers who helped package and label everything were tremendous. In terms of getting our lab CLIA certified, Sarah Dry, Alyssa Zidman, Linda Baum, oh my, tremendous partners, and we're very grateful. And, you know, we not only do these assays, but we have to report them out to the participants as well as to LA County, to the state, and to DHHS. So just in terms of participants, we see on average 200 a day. That's 400 emails that have to be sent securely. Sending results to the state and to DHS has just been a tremendous lift. Just so you realize, like, Pasadena is in LA County, but it's a separate health system. But even within a zip code, some streets belong to Pasadena, others to LA County, and we have to sort of sort that out. And we've worked on bioinformatics and getting through all the Part 11 and all the regulations have been tremendous. And so we thank Michael and Simon very much. We thank the postdoc union, and I won't get into that. And then in terms of our firefighters, I can't say enough good things about Chief Brian Kane, who has been just an extraordinary partner, and the fire department medical officers, Clayton and Saman. It's an ongoing, it's a true partnership, and we learn, and we're so honored to be working with them and to help them in their tremendous efforts. And again, I can't mention the firefighters without mentioning Ashley and Rachel, because they're spending 3 days a week out there in the blistering heat, getting everything ready. Again, doing everything that it takes to stand up this study. So I think there's a little bit of time for questions. And one final thought. So thank you for your attention. And I don't know if anybody's there.
Great, thank you so much, Grace. That's really tour de force. I don't think any of you guys sleep. All right.
I've made chocolate and wine.
Could I jump in?
He has a question. Yeah.
I just have to say, Grace, that it's an extraordinary presentation, extraordinary amount of work, and so important. And I want to congratulate you and everyone that whose work you presented, and especially the teams behind you. I was thinking that having established such a complex and functional group, have you had any thoughts about how you might be using this sort of infrastructure to look at the time of vaccination?
Thank you for the compliments, Gay. I'm part of a team and incredibly honored and privileged. Yes, and so we, when we get positives, we will refer them to studies that Judy Courier and Rafi and Risa and others are doing. And it'll be really interesting. I had thought that the firefighters would be open to vaccinations, not really. But we'll see. It's an ongoing conversation. In terms of, understanding immune response and how that might inform vaccine studies, yes, that's something that we're just starting to do. I will say that none of our participants who have become infected while we've been following them have had severe disease or moderate. At most they've had fever and stuff, but we really are hoping, particularly since we have pre-infection samples on them, that our studies will be able to inform vaccine efforts. We are only now at the point that we can breathe a bit to actually do science. Up until now, we have been, every moment of the day has been consumed with overcoming challenges, labeling tubes, processing samples, but we now have enough toys, if you will, that we can start doing the fun stuff, which is why we did this in the first place. So I don't know if that answers your question, but.
Yeah, it does. Thank you, Grace. Yeah, it just, there are so many aspects to the whole topic of vaccination, and you touched on one of them is interest and compliance with vaccine strategy, and you've already got some insight as to how that's going to be difficult, but also sort of who should get vaccinated based on your data, and how do we then follow those results. And I guess actually the vaccination trials are going to all have that built into them, aren't they, as to how immune responses are evaluated. I don't know how much can be done around the fringe, if you like, beyond those clinical trials.
Oh, I agree. And they're very well-designed clinical trials with a lot of important science. I think another advantage that, though, of our cohort is this whole issue of reinfection. We know for the, seasonal or human coronaviruses that reinfection is the rule. I have to say that, I was surprised that so early on we're already seeing cases of reinfection. I mean, documented by sequencing viruses at 2 time points. I think now there's 7 cases. I haven't checked the website. There's a website for everything now, right? The last time I checked last week, there were 7 documented cases. Now, you could argue that that's just because everybody is studying this so intently, but it does give pause. And as you know, Gay, just because you can detect neutralizing antibody doesn't mean the vaccine's going to work. There's so little, I mean, we've learned a tremendous amount, but I want a vaccine. But I'm not sure, and we'll have to see. And I think that's why it's so important that we try the vaccines that we do, and that they really be well conducted. And I was heartened that pharma has said that they're not going to cut any corners. But as you know, AstraZeneca's vaccine was halted. Studies were halted yesterday because of a case of transverse myelitis. And one of the things that everybody is, well, not everybody, but a number of people have been concerned about is enhancing antibody or other things. So we'll have to see. We don't even know if that case was in a placebo, but it's interesting times.
Thanks, Grace.
Oh, thank you.
Hey, can I ask a question?
Go ahead, Fong.
Yeah, okay. So you mentioned that the COVID-2 RNA is detected in breast milk. Do you know what form those RNA molecules are in?
We don't now, but Paul Krogstad, who's been working on this, has developed a PCR to look at all the different species of RNA, and we're hoping that actually that will avoid having to do culture. But so stay tuned. As of last week, Paul's on clinical service now, but last week I've not seen the data, but he was happy with his PCR. So we'll learn.
All right, great, thank you.
Okay.
And Grace, I guess related to Feng's question, I'm curious, do you think that the RNA you detect in the breast milk could potentially prime the infants before the infant is infected by the mother?
That's interesting. I don't really think that there's enough there, and it would be a difficult question to address in humans, but yeah, so when antigens get presented in milk, it does tend to tolerize infants. So Native American people would give poison oak and poison ivy to the infants under the cover of, while they were breastfeeding, so they wouldn't react. So that's a interesting question that maybe we can, once we get through this insanity, we may be able to look at.
That's cool. And I think our last question is in the chat room. How many pregnant patients are in the healthcare worker study?
Oh, pregnancy we don't exclude. I know one maybe one pregnant woman who actually became infected while on the study. I remember her well because I do her blood and stuff. I want to say there are 2, but I don't know for a fact.
Grace, it's Kathy. There are 8.
8? Oh, geez. Okay. So we have 8 pregnant women.
Great. Well, thank you, Kathy. Well, thank you very much, Grace. I think there are no more questions. And thanks so much for, you know, a fabulous update, and everybody get to hear about the, you know, impressive work that's going on here. And next week, we will see everyone same time on Wednesday. Thank you, everyone. Bye-bye.